Hey guys, so in this video we are going to do the histology of the reproductive system. So we have the two different organs to check out and look at. Start off with the male gonads. We have testis, and so we've got our image. Thank you, OpenStax, for this to show you where our histology is coming from. So we've got the tunica albuginea is kind of like this hard tunic that holds the seminiferous tubules. And the seminiferous tubules are where the sperm is made inside of the testes. So if we zoom in and get to our slide now, these right here are the seminiferous tubule. So here is one tubule, for example. On our tubule, we are going to have our germ cells that make the sperm. So we are going to have the spermatogonium. That's these guys on the edges. The spermatogonium are going to be our stem cells that are going to make the sperm part of our germinal epithelium. These guys are going to produce little baby sperm called spermatids. And these guys, after meiosis of the spermatogonia, after they undergo meiosis, we end up with four little unique baby spermatids, and those guys will make their way to the lumen here, to the inside. Once they make it to the inside, then they have to travel all the way out of the testes and kind of hang out in the epididymis where they grow up and mature for a little bit until they're needed. So those are the tubes, the seminiferous tubules right here. We got some Sertoli cells. We got some little myeloid cells over here near the periphery of the tubules. Other cells that we need to know for sure are the Leydig cells. The Leydig cells are oftentimes called interstitial cells too. And so the interstitial cells, Leydig cells, those guys are going to produce the hormones associated with males, which would be testosterone and inhibin. If testosterone is going to be our male associated hormone, then inhibin would be the one that comes along when those levels get too high to shut that process down. Inhibin would shut it down. So those two hormones are coming from these guys, these cells. So these are the Leydig cells. They're kind of squished between the tubules. That's why they're called interstitial cells. So if we've got a tubule here and here's another tubule, it's usually those little triangle-shaped cells that get squished between the tubules. So those are going to be our Leydig cells, interstitial cells producing testosterone and inhibin. These are the whole tubules. They are lined with spermatogonia that are going to undergo meiosis, meiosis 1 and meiosis 2. And at the end, they produce little spermatids, and those guys will make their way into the lumen, and they will travel all the way out of the testes and into the epididymis, where they will finish growing up and maturing. That is going to be our basics of the testis histology. Then we have histology for females. We have the ovary. And so I want to show you the parts of the ovary on this organ. It also has a tunica albuginea that goes on the outside. So this kind of hard-ish tunic kind of wraps around and holds everything in place, tunica albuginea. And then we have this succession of development of eggs, the little oocytes, and then little eggs, the little oocytes are going to be wrapped in follicles, and those things will grow and develop. So all of the eggs are going to be hanging out, born in this area along the edge of the ovary. So I hope you can make out that the edge of the ovary is kind of lighter colored. And the lighter colored section is the primordial follicles. We're going to go into each one of these in just a minute. But I just like to show you the whole ovary and that you can see everything all in once, mostly on just scanning power on your ovary. It's pretty cool. So those are the primordial follicles, the baby eggs, little primary oocytes surrounded by primary follicle, primordial follicles. Then we go to a primary follicle, then we go to a secondary follicle, and then we go to a mature or tertiary, aka Graafian follicle. So we will need to be able to identify all of these follicles that progress after puberty hits and those follicles start to develop. We need to know what the different follicles are, what they look like. So other things on here, we've got our germinal epithelium. Again, that's just the place where we are going to have the oogonia they were here, and they were going to produce all of the little primary oocytes that we are born with, that is happening in utero. And we've got all the eggs that come out inside, as primary oocytes inside of primordial follicles on the edge. And then we talked about cortexes and medullas when we saw that slide of the kidneys. So we've got cortex on the outside, medulla is going to be on the inside. So we've got ovary cortex here, and then internally it would be a medullary area on the inside. So going through the follicles, we have our first stop is the primordial follicle. And again, I said it was on the edges. So here is that tunica albuginea on the outside, and all of these little things that I think look like bubbles. These are all little teeny tiny little eggs called oocytes, and they are the first stage. So egg development is going to go through meiosis just like sperm development does, but there's some differences. The eggs are all going to stop at prophase 1. So at prophase 1, we went through meiosis. First stage is prophase. The little homologous chromosomes have made sisters, and those guys have aligned up. So now we have homologous chromosomes and their sisters, and they line up and they make a tetrad, and we end up having crossover happening at prophase 1. And that's why all of the eggs are different. That's why all of the sperm are different. So these guys are primary oocytes. So these are primary oocytes, which means they are still stuck in prophase 1 and they have not developed past that as primordial follicles. These primary oocytes, these little bitty baby eggs, are going to be surrounded by a little bitty little baby follicle, the primordial follicle. The follicle is basically like the bag, the sac around the egg. So on our primordial follicle, these guys, if we zoom in, you can make out the little edge, the follicle edge. So these are going to be little simple squamous, real thin cells that go around. This is a primary oocyte. Here's another primary oocyte. Here's another one stuck in prophase 1. Then puberty hits. Puberty hits, at different ages for different people. But once puberty hits, your tolerance for the sex hormones, for testosterone and for estrogen, start to go way up, a lot higher than they were before puberty. So you start getting all the secondary sex characteristics that happen with those hormones, and you also develop the eggs. So we are going to go from a primordial follicle into a primary follicle into a secondary follicle, and we are going to go from prophase 1, and eventually we will make a secondary oocyte that stops at metaphase 2. So we are going to start to go through the rest of meiosis and stop at metaphase 2. So our next stop after primordial was primary follicle. So our primary follicles, we still have our little primary oocyte. Here's our little egg, and it is going to be surrounded by the follicle. And in this case, check out this follicle. This follicle is 1 layer thick, roughly. We're starting to sneak in a second layer here, but for the most part, 1 layer thick. And it is, look at those cells. Those cells are not pancake anymore. They're cuboidal. They're about as wide as they are tall. So these guys, cuboidal cells. We got cuboidal cells going around the edges of the follicle. So those guys are our granulosal cells. They are simple cuboidals. Simple cuboidal granulosal cells on the outside. And then our primary is going to keep developing. So we're going to go from simple cuboidals to stratified, lots of layers of cuboidals, lots and lots of layers. So look at our secondary follicle. This follicle has gotten bigger and bigger around the egg. Look how much thicker it is. So we're getting for sure a whole second layer up here. We are getting stratified. We are starting. Over here, much more stratification going on. Look at all these layers over here on our secondary follicle. This, again, is our oocyte. It's our little bitty egg in there. And it is also starting to develop this space. Look at this space, this white stuff. It's filled with fluid. And this little space here is called an antrum. That's the antrum. As the follicle develops from a secondary follicle to a tertiary or Graafian follicle, the antrum gets very well defined and clear. At the secondary stage, we are just starting to form an antrum. So that's the giveaways for secondary. If you see a slide and you're wondering what stage follicle am I looking at for this egg development, what follicle are we at? we are at a secondary follicle because we have multiple layers of the cuboidal granulosal cells on the outside. They're starting to differentiate. We are going to start to differentiate into theca cells and granulosal cells. On this follicle, we are going to very much have all these layers, and we start to develop this. We start to develop this fluid-filled antrum beginnings. It's not totally perfect yet. We're still at a secondary follicle. The last stage is going to be our tertiary, aka Graphian, aka mature follicle. This is the best it's going to get. So by this time, our little egg in here has now gone through prophase 1, metaphase, anaphase, and telophase of meiosis 1. Then they go into meiosis 2, prophase 2, metaphase 2, and they stop. So this is a secondary oocyte stuck in metaphase 2. Look at this follicle development. That follicle looks beautiful. It is very large, big, very much stratified, lots of layers in there. Look at this nice, clear, well-defined antrum on this. This is a mature Graphian follicle. Then check out the egg too. So on the egg, it is going to have layers. So here is our oocyte. Here's our little ovum. And again, it's a secondary and it's stuck in metaphase 2 of meiosis. We need to know the layers outside the egg because even outside the egg has developed now that we are grown and mature. So this has this light colored area. I don't know if it really comes through properly on our imagery, but under the microscope, this area is a light pink. It's light pink staining, and the egg itself is a little more clear color. But the light pink area, this area right here, is the zona pellucida, the ZP. The zona pellucida is immediately around the egg, touching the outside of the egg. And then outside of the zona pellucida is a whole nother layer, and that layer is called the corona radiata. So it's literally like a radiating crown. That's what it means. It's a crown that radiates out. So that's all these cells out here. So all this purple stuff, this is the corona radiata. All of this corona radiata. So definitely be able to recognize the egg is here. The ZP is immediately around the egg, and then around the ZP on the outside of the egg is the corona radiata. That's the last part. And this, again, we said is a mature Graphian follicle. It is surrounded by our granulosal cells, our theca cells. Those guys are going to be secreting different hormones, estrogen, progesterone, inhibin. If things get too high, we can inhibit and shut things down. It can also release activin. So here is our granulosal cells all here. When we are on day 14 of the ovarian cycle, this follicle is going to rupture and burst. So this follicle will burst on ovulation, day 14, and that egg right here is going to come out, and eventually it will pop out and it will go into the uterine tube, fallopian tube. It has like 3 or 4 different names out there, but it'll be caught. We're gonna have little fimbriae that attach on, and they are going to catch it like a little catcher's mitt, catch the egg, and it will go through the fallopian tube on ovulation. So this is very much well developed. When that egg pops out, then this follicle, all of this theca granulosal cells, these guys are going to take over and they will become the corpus luteum, this yellow-like substance that's going to secrete progesterone for a future pregnancy because the egg is out there, the egg's viable for 24 hours, and if it meets up with some sperm, it gets fertilized, pregnancy could happen. So this follicle will then come along and secrete progesterone and help support the future embryo that should implant about 3 days later in the uterus. If that does not happen, the egg just keeps on going throughout the body, and then the corpus luteum changes from yellow color to white, and it is corpus albicans if that egg never got fertilized. If the egg did get fertilized, it is going to go from meiosis to metaphase 2 through to anaphase and telophase, and it will complete its process of meiosis, but only completes meiosis if it gets fertilized. Otherwise, we just have the secondary oocyte, and it is always stuck in metaphase 2 in meiosis. So these are the histology For the ovary, it's a little more complicated than testes. It is, but we need to be able to identify all the different follicles and any special features about those follicles. What type of epi is on the outside? What is surrounding this stuff? So we got simple squamous here. We have simple cuboidal here, and then we get stratified. All of this is stratified cuboidal on our secondary and tertiary levels. So I hope this helps you get through the reproductive slides to understand what that histology is. And have a wonderful rest of your day.